So I'm a geneticist and anthropologist. Really, my training is in what we call human population genetics. Now, what do human population geneticists do? Well, the overarching theme in all of our work is to try and explain the patterns of human diversity we see when we look around the world. We travel, we walk down the street in a major city, we see people who seem to be so different from each other and from ourselves. How do we explain these patterns of diversity? That's really the overarching theme of our work. Let's rewind through time, a couple of hundred years. Imagine you're setting sail during the European Age of Exploration with Captain Cook, headed out to the Pacific, maybe looking for Terra Australis, the mysterious continent that kept the Earth balanced on its axis, otherwise it would have fallen over like that, or at least that was the thinking in those days. You're setting sail and you come ashore on all these islands along the journey, and you find people living there. You're not surprised at the fact that there are humans there, although in retrospect it is a little bit surprising how far people went out into the Pacific. And they speak a language that's somewhat like other languages you've heard. They look a little bit like other people you've seen nearby, but somewhat different. How did those patterns of diversity arise, and how did people get to every corner of the Earth? That's really the question we're trying to answer. And like any big overarching quest in science, you can break it down into sub-quests or questions that you can start to chip away at using the tools of science, gathering data, formulating hypotheses, coming down on one side or the other. And the first question we can ask is, looking at this panoply of human diversity, are we in fact all related to each other? Can we trace our origin as a species back to a single source? A question of origins. And the other question we can ask is one of journey. If we do spring from a common source as a species, how do we come to populate every corner of the world, in the process generating these patterns of diversity that we see today? Well, the question of origins, as with so many other big questions in the study of biology, seems to have been answered over a century ago by Charles Darwin in his second most famous book, so not The Origin of Species, but The Descent of Man, published in 1871. He wrote, in each great region of the world, the living mammals are closely related to the extinct species of the same region. It's therefore probable that Africa was formerly inhabited by extinct apes closely allied to the gorilla and chimpanzee. And as these two species are now man's nearest allies, it's somewhat more probable that our early progenitors lived on the African continent than elsewhere. Okay, so we're done with the origins question. We can wrap this up and head on out for an early lunch. Not quite. Darwin, of course, is talking about our shared ancestry with apes in this statement. He was absolutely right, even though he had no real data to support his hypothesis at the time. Apes do actually first appear in the fossil record in Africa around 23 million years ago, at a time when Africa was actually disconnected from Eurasia, floating around the Indian Ocean due to the vagaries of plate tectonics. It bumped into Eurasia between 16 and 18 million years ago, and at that point we had the first so-called African exodus of apes. The apes that left at that time, some of them ultimately ended up in Southeast Asia, evolving into the gibbons and the orangutans. And the ones that stayed on in Africa ultimately evolved into the gorillas, the chimpanzees, and yes, us. So yeah, Darwin's absolutely right. If you go far enough back in time, 23 million years, we as a species share an origin in Africa along with every other species of great ape. But that's not the question I'm really asking. Rather, I want to know, what about us as a species, Homo sapiens, things that we would recognize as being like us if they were sitting out here in the audience? Where did we come from, and when? Slightly different question, and one that historically has been approached through the study of bones, paleoanthropology, going out and digging things up out of the past and guessing about how they may or may not connect up to the present. What I'd like to suggest as a geneticist who deals in large quantities of data is that the field of paleoanthropology has relatively spotty data, and therefore, instead of giving us probabilities about our ancestry and our origins, it gives us possibilities. Possibilities, not those probabilities that we as scientists, driven by data, at least, really want. Now, what do I mean by that? This is a great example. What you're looking at here are three potential human ancestors: extinct species of hominid, from left to right, Homo erectus, Homo habilis, and Paranthropus boisei, a robust Australopithecine, say that 3 times fast, all uncovered from the same location on Lake Turkana in Kenya, actually by the Leakey family, and all dating to roughly the same time, about 2 million years ago. So we've got 3 potential ancestors living in the same place at the same time. That means not all 3 could have left descendants down to the present day and to me. Which one of these individuals am I related to, if any? I don't know. Possibilities about our origins and our past and our ancestry, but not the probabilities that we're really looking for as scientists. Well, we as geneticists take a slightly different approach. Instead of digging things up out of the past and guessing about how they may or may not connect up to the present, we start in the present and we work our way back in time. Because we know that everybody alive today had parents, by definition, and those parents had parents. So in principle, it is possible to build a family tree for every living human being today. It's a genealogical approach. It turns the effort on its head. Instead of guessing at things that may be related in the past, we start in the present, we work our way back in time. Now, I'm sure there are a few of you in the audience who are genealogists, have built family trees. How far back can you go? A couple of generations, a few hundred years? But what happens eventually? You hit what the genealogists call a brick wall, a point where the written record runs out, and beyond that we simply enter this dark and mysterious realm we call history, and ultimately prehistory. But it turns out we're all carrying what is in effect a historical document inside of ourselves, inside of nearly every cell in our body, in our DNA. And this was actually a still image that was shot on the day that we were filming that street fair in Queens that you just saw some images from in the film. Now, DNA, a quick primer for those of you who have not taken molecular genetics recently. You know who you are. There will be a quiz at the end. Long linear molecule, the famous double helix described by Watson and Crick back in 1953, for which they later won the Nobel Prize, composed of 4 subunits. We denote them A, C, G, and T. And it's the sequence of those A's, C's, G's, and T's that essentially provides a blueprint to make another version of you. And there are a lot of these nucleotides, as we call them, in the human genome, billions of them. And so the DNA molecule is very, very long. How long? Well, if you took the DNA out of one tiny microscopic cell in your body and you stretched it end to end, it would be about 6 feet long. If you took the DNA out of every cell in your body and stretched that end to end, it would reach from here to the sun and back about 300 times. So it's a lot of information. And in every generation, you have to copy all of this information to pass it on to your kids and your grandkids and so on. And it's really important stuff. Again, it's the blueprint to make another version of you. But it's like copying the longest text imaginable. Think of a long book, War and Peace, let's say. It's like 1,000 volumes of War and Peace. And you've got to copy that by hand in 8 hours, which is how long it takes your DNA to replicate. So what are you going to do? Well, you're going to sit down with your Red Bull or your coffee or your beverage of choice, and you're going to go back and forth and copy and check and double check, proofread this text as you're copying it to pass it on, because it's such important information. But inevitably, what is going to happen during this process? You're going to make a typo. Those happen at the DNA level as well. We call them mutations. And when they occur at a very low but a measurable rate of around 100 mutations per genome per generation, as the DNA is being copied and passed on, as they occur and get passed down through the subsequent generations, they become markers of descent. If you share one of these markers, one of these changes in the DNA sequence with another person, you share an ancestor, that person in the past who first had the change in their DNA and passed it on to the two of you. Now, what do these look like? Well, this is actual DNA sequence data off of an automated sequencer, slightly dated these days. We use a little bit different machinery, but the old-fashioned Sanger sequencing is still kind of the gold standard, and it's easiest to see what these changes look like in this form. These are 5 people, 1, 2, 3, 4, 5, who've had the same region of their DNA sequenced, lined up. And the first thing you'll notice as you read down through these sequences is that they're basically identical. That's the first thing that comes out of all of our studies of human genetic diversity. Humans are essentially identical at the DNA level. We're 99.9% identical. In fact, we only differ at, on average, 1 in every 1,000 nucleotide positions from someone we're not even related to. So finding these markers is a challenge in itself. It's like finding that needle in the haystack. But if you look carefully enough, down here in this region, GGG, GGG, GA, G, a single letter change from a G to an A, that's an example of a marker. If you share that with somebody else, you share an ancestor, the person who first had that change in the past and passed it on to the two of you. And it's by connecting those genetic markers up in that way into shared ancestry that we are able to build family trees for everybody alive today. And you saw in the film, talked about mitochondrial DNA, the Y chromosome. This traces a purely female line of descent. The equivalent on the male side, fathers pass it on to their sons. By looking at the markers, we're able to build these family trees. Everybody in this room, in fact, everybody walking around on planet Earth today, falls somewhere onto one of the branches of these family trees. It's kind of cool. Now, these are actually incredibly simplified versions of the trees that we use in doing the laboratory analysis, but they're still kind of complicated, sitting out there a little after 11 in the morning. So let's simplify them even more. What's the take-home message here? Combine them, turn them on the side so the roots at the bottom, the branches are coming off the top, like a regular tree. Well, it's that the longest branches, aka the deepest branches in the family tree, are found in Africa, only in Africa. And because the length of these branches, again, is proportional to the number of those mutational changes ticking off in a clock-like fashion, 100 per genome per generation, what that means is that Africans, as a group, have been accumulating genetic diversity for longer than any other group of humans. And therefore, our species originated in Africa. And at some point in the past, a group of Africans left that continent to go out and populate the rest of the world. Okay, well, we're back where Darwin started. We've just wasted a ton of time on complex DNA stuff. Except we haven't, because we're not talking about an African origin 23 million years ago. Rather, we're talking about an origin within the last 200,000 years. That's a few thousand human generations. When geneticists made this discovery in the late 1980s, early 1990s, the discovery of mitochondrial Eve, it was earth-shattering to people in the anthropological community. If you had taken physical anthropology as recently as I did back in the 1980s, you would have been taught that human races had been diverging from each other, so Europeans from Africans, from East Asians, for millions of years, back to the time of Homo erectus, perhaps. This was really earth-shattering to the anthropological community. We're so closely related as a species. Moreover, according to the genetic data, we only started to leave Africa within the last 60,000 years. That's only 2,000 human generations, literally the blink of an eye in an evolutionary sense. Scattering to the wind, a coastal migration leading very rapidly to Australia. It's been settled for at least 50,000 years. There was a paper that came out on that just a couple of weeks ago, one of the oldest populations on Earth, certainly the oldest outside of Africa as far as we can tell. Later migrations inland through the Middle East, Central Asia, moving into Europe by around 35,000 years ago, and a small intrepid band headed into the worst part of the last ice age during the last glacial maximum between 15 and 20,000 years ago, crossing a short-lived land bridge, the Bering Land Bridge, between the Americas and Asia, and populating the New World. The great Paleolithic wanderings of our species. And learning the details of these migrations and the how and the why, the timing and correlating with archaeological patterns, linguistic patterns, and so on, is what I've spent my life as a scientist focusing on. And it's what we focused on in the Genographic Project, which I founded in 2005 with National Geographic and IBM. We're doing, obviously, at its heart, it was field research focused on working with indigenous and traditional people living around the world. Now, why are indigenous groups so important to this work? Think about your own ancestry. I'll think about mine out loud. I have ancestors from all over Northern and Western Europe. I live right now in Texas, have lived at various points in other parts of the world. What does my DNA tell you about the ancient history of any of these places? It's hard to tell because I'm a mutt. In the last 300, 400 years of migration. has really mixed up most people's genomes in the world. But there are people who retain a link back to their place of origin that goes beyond a few hundred years, back thousands or even tens of thousands of years. And these are the world's indigenous and traditional peoples. They give us an insight into those ancestral genetic patterns that we need to track the ancient migration routes. But when we were designing the project, and this is kind of the cool, unexpected thing that came out, I felt very strongly that it shouldn't just be the story of the world's 100, 200 million indigenous people. It's the story of all of us. And I've been asked so many times over the years as we have published papers, hey, could I get my DNA tested to find out something about that? And you couldn't, really, until we decided to include what we called the public participation component in Genographic, an opportunity for anybody who's interested to send in a DNA sample, covering the cost of the testing, of course. We can't afford to sequence literally everybody's DNA on Earth, but we wanted to give people an opportunity at a reasonable cost to find out something about themselves and also join the project. So the public participation component, a really vital part of what we did in the project. By selling these kits, it's all nonprofit, everything got plowed back into the research effort and into the third thing, tangible, back to the world's indigenous and traditional peoples, many of whom lead extremely marginalized lives in already desperately poor places. And they're often forced to leave behind their ancient villages, their homelands, typically moving to a growing megacity. Think of Sao Paulo or Mumbai. And when they do, they become part of the melting pot. Their kids stop speaking the original language, and that culture effectively dies. We are going through a period of cultural mass extinction at the moment that parallels the biodiversity crisis. Linguists tell us that of the 6,000 some odd languages spoken around the world today, by the end of this century, between half and 90% will be extinct, no longer spoken, gone forever, and most have never been written down. We, as a team at Genographic, felt very strongly that human diversity is what really defines us as a species. We don't have that much going for ourselves physically. We can't run like a cheetah. We can't fly like a bird. We don't have warm fur like a snow leopard, but we can invent cultural ways of doing all of those things and of going to the moon and sending a spaceship to Pluto and sequencing our own genomes. We're defined by our diverse culture, by our intellect. And when we lose a culture, we lose a piece, a chapter, if you will, in the book of what it means to be human. So these were grants that we created, applied for by the groups themselves to preserve cultural diversity around the world. Now, just a quick glimpse at the numbers. Over 72,000 people from indigenous groups, over 1,000 different populations around the world on every continent joined the project. So far, we have over 50 publications. I think we're actually closer to 60 now, and many still coming through. We have a paper that's under submission right now about the genetic history of Chad, an expedition I led several years ago where we drove up to the Tibesti Mountains, right up on the Libyan border. Fascinating trip, the first modern expedition to go there because it's in rebel-held territory and surrounded by landmines, so not the easiest place to get to, but revealing details about how people migrated across the Sahara. The big surprise that came out of the work that we were doing is how excited people were about testing their own DNA. When we were putting this component of the project together in 2004, before we launched in early 2005, I actually had a bet with the then CEO of National Geographic, John Fahey. John said, Spencer, I love the idea of the project, I get the science, but I gotta tell you, nobody's gonna pay $100 to test their DNA. Nobody cares about that. If you sell 1,000 of these things by the end of the project, which was initially intended to be 5 years, I'll give you $1. John was a big spender. I said, okay, you're on, John, because I really do think that people are interested in this stuff. So we launched in April of 2005. On the first day alone, we had 10,000 orders for DNA testing kits. We hit 100,000 8 months later by the end of the year and continued to sell between 50 and 100,000 kits from then on. And it really created this modern industry of consumer genomics, and I'll touch on that again in the end. People from more than 140 countries, too. Most of the sales are in North America, Western Europe, Australia, but we've had orders from Uzbekistan, Kazakhstan, 2 from Vatican City. Really love to see those results. And it also raised a fair amount of money, not only for the research, but also for the grants. We gave away over $2.5 million in legacy grants. And just to give you a sense of what these legacy grants were all about, A project that we funded with the Yagnobi people of Tajikistan. They historically lived in a remote river valley, the Zarafshan Valley in the northern part of the country. Most of them were resettled during Soviet times and later in the 1990s by the Tajik government into the capital, Dushanbe. This is a language that's spoken by maybe 1,500 people. The kids don't learn to speak it. They go to school in the dominant languages, Russian and Tajik. And the people who applied for the grant said, listen, our culture is literally on the brink of extinction. We're not going to have this language anymore. The kids aren't going to know anything about who we are as a people. So we helped them fund school curricula in a few subjects to give the kids a sense that it's worth learning this language, worth keeping it around. That's the main reason a lot of immigrant kids stop speaking the original language, because it's cool to be part of the dominant culture. You don't want to speak the language your parents did. Well, in this case, it shows the kids that you can actually learn useful things in that old language that their parents and grandparents speak. Now, why is Yagnobi important? Some language spoken by a handful of people in Central Asia. Well, if you were able to rewind about 1,500 years, this would have been the lingua franca along the Silk Road. If you were trading goods anywhere from the Caspian to the ancient Chinese capital of Xi'an, in places like Bukhara and Samarkand, you would have been trading in that language. So if we lose that language, we lose an insight into that very important chapter in human history. A project in the Northern Territories of Australia to preserve Aboriginal dance patterns along with their song lines, their stories of origins, which very interestingly seem to correlate somewhat with the genetic patterns. A project along the Yukon River in northwestern Canada and southeastern Alaska, raising awareness about environmental issues that are threatening the salmon stocks, and everybody's life revolves around salmon fishing up in that part of the world. And this is an interesting project for people who say, well, surely this is just social Darwinism writ large. Some cultures are meant to be, in the ascendant, and others are going to be left behind, and it's just the way of the world. And why does it really matter if we save all of these cultures and languages? This is a project that we worked on. It was actually one of our bigger grants. We gave about $100,000 for this, to try and preserve the ethnobotanical knowledge of the Shuar people living in the foothills of the Andes in Ecuador. Now, if you need a pragmatic reason for why it might be worth saving some of this information, think about medicines you might be prescribed by your physician. Ultimately, between 30 and 40% of those trace back to plant sources. And we often know about those plant sources because of accumulated traditional knowledge over thousands of years, times thousands of populations. So when we lose that knowledge, so hard to acquire, so easy to lose, when we lose that knowledge, how many potential treatments for cancer or HIV or Zika might we be missing out on? We don't know, but it's worth preserving. So just some examples of what we did with the legacy grants. Now, as I said, the most surprising thing to come out of the project, apart from some cool scientific findings, obviously, was the excitement on the part of the general public in getting themselves tested, and the way that that can feed back into the research. I like to think of it as the power of what we call citizen science. And this is a great example. It came to light by accident a few years ago. A woman wrote in to the project and said, "Dear Genographic, love what you guys are doing. Think it's fascinating. I've found out all sorts of interesting stuff about extended members of my family. Lots of people have tested. We've given the kits as gifts. However, in my case, I think you got the results wrong. And I'd like you to retest me, because you told me that I'm carrying a Central Asian or Siberian genetic lineage. And I can tell you for a fact, in fact, I can show you church records going back to the 16th century, that I came from a little village just outside of Budapest, which is what you're looking at here in Hungary. So clearly you got it wrong. I'm European. I know that. Please retest me. Thank you very much, et cetera, et cetera." Now, when I read this email, I got really excited. Not because I enjoy asking the lab to cherry pick one sample out of hundreds of thousands. They tend to get very annoyed at that. But rather because the Hungarians are a really interesting group in Europe, at least linguistically. Most of the languages of Europe fall into what we call the Indo-European language family. So the language I'm speaking now, which is part of the Germanic branch, so German, also a member of Indo-European. The Romance languages, French, Italian, Latin, also Indo-European. Russian, languages spoken down in India, like Hindi, Farsi, spoken in Iran, a Western Eurasian group of languages. And most of the languages of Europe, as I said, belong to this language family. Very interesting story. It seems to have come in from the steppes of Central Asia during the Bronze Age. That's not the story we're telling right now. What is interesting is there are a couple of languages in Europe that don't fall into that family. One is Basque, which is an outlier. It's a linguistic isolate, as linguists call it. It's unrelated to anything else on Earth. It could have been brought here from Mars, as far as we know. It's spoken in northeastern Spain, southwestern France, and it's not related to anything else, as I said. And then the other outlier is Hungarian. Hungarian spoken down here in the Central European plain, and it's related to languages spoken up in Eastern Scandinavia, Northern Eastern Scandinavia, like Finnish, the Finno-Ugric branch of what's actually called the Uralic language family. And as the name Uralic suggests, the center of diversity is in the Urals and points further east in Siberia. Now this makes sense because we know from written sources that a group of steppe marauders, the Huns, or Magyar people, migrated from the steppes of Central Asia into Central Europe about 1,000 years ago, conquered, brought with them their culture, their love of chicken paprikash, and actually that came later, the peppers came in from the Americas in the last 500 years, but their language. And so they had a huge cultural impact, complete shift in language, replacement of whatever was spoken there before. And clearly, with that sort of a replacement, you must have a genetic impact as well. The problem is, when we've gone in and done typical studies that we do as scientists, sampling, say, 100 people, the Hungarians look identical to the surrounding groups, the Poles, the Slovaks, etc. So, how could there have been such an outsized cultural impact? Well, when this woman wrote in, I said, this is an opportunity to make use of this growing database. I wonder how many people in there have declared Hungarian ancestry. So going back, mother, mother's mother, mother's mother's mother, language spoken Hungarian, etc. Cleaning up all the data, we were able to pull out 2,300 people at the time that we did this analysis a couple of years ago with known Hungarian ancestry. And what we found was that we do see these Central Asian or Siberian genetic lineages on both the male and the female side at a very low frequency, 2 or 3%. This is the power of large quantities of data, which is what we knew about going into the project. What we didn't anticipate is that the reason we looked for this pattern is because this woman was curious about her own results. That's the power of citizen science. Instead of having a few people with PhDs deciding on the questions they want to ask, decentralize it. Give the power to hundreds of thousands of people who are all driven by curiosity about their own result. Give them the tools, and they will help you discover really cool stuff. So that's something we've built into the consumer genomics platforms that we're developing right now and will continue to develop in the future. I want to talk a little bit about two big revolutions that have happened in the last few years in the field, going beyond the Y chromosome, mitochondrial DNA, and really kind of taking the field into the future in a new way with technologies that we really didn't anticipate having access to back when we launched the Genographic project. The first revolution is the revolution in whole genome sequencing. And this is the ability to not just sequence segments of DNA, but literally all 3 billion nucleotides in the human genome, generating massive quantities of data. Totally unanticipated when we started the project. Now it's routine. And the other revolution is in ancient DNA. The ability to extract DNA from ancient remains, bones that have been buried for tens of thousands of years, giving us insights into stuff that went on in the distant past, literally a time machine. Instead of using complex statistical models and data from people alive today to guess at what might have happened in the past, you can literally go back in time and be there on the ground with these people, test their DNA, really get at the tempo and the mode of evolution. Well, the revolution in whole genome sequencing is really unprecedented for any technology in human history. Those of you who work in the computer science field will know about something called Moore's Law. Gordon Moore formulated it not so much as a law, but an observation in the late 1960s, early 1970s, when he noticed that microprocessors seemed to be getting quicker and quicker, And so if you waited a couple of years, you would be able to generate the same amount of processing power at roughly half the cost, or if you were spending the same amount of money, you should expect a doubling every 18 to 24 months in the processing power. Very, very rapid technological change, and it has held up over the course of the last several decades. Sequencing was basically following Moore's law. If we go back to the first human genome, the Human Genome Project that you've all heard about, it took over a decade. It cost $3 billion. It involved scientists from around the world, a private company called Celera, led by Craig Venter. The announcement of the draft, June of 2000, by President Clinton, with Craig Venter and Francis Collins flanking him in the West Wing of the White House. Big, big, big announcement, the very first human genome to be sequenced. Hugely expensive, complicated, took a long time. Now you can do that in 3 days for less than $1,000. That's unprecedented. This, what we call next-gen sequencing, which really took off in about 2007 when a company called Illumina acquired another company called Solexa, perfected the technology, and essentially what they do is they sequence tiny amounts of DNA bound to microscopic slides, and they do it in parallel at millions of positions around the genome. And then you use computers. It's incredibly complex computationally to then assemble a genome, but they use computers to assemble the final genome sequence, making use of the fact that we have other genome sequences that have been completed before to compare to. So the drop in cost has been precipitous, and it's asymptoting towards zero. So DNA sequencing is going to be essentially free in the next few years, which raises a question about, what happens then? And I'll talk a little bit about that in a minute, but for our purposes, it gives us an incredible tool for understanding the details of human evolution. The Y chromosome, mitochondrial DNA have been really important for understanding the who, the where, the when. But the how and the why, we always have to compare to other fields, archaeology, linguistics, and so on, and look at correlations. And sometimes those correlations are correct, the inferences are correct, and sometimes they're way off the mark. What if we could use the DNA sequences to objectively tell us about the how and the why of human evolution? What makes us human? What's been selected for and against during the course of the last 200,000 years? Well, it turns out you can make use of the system itself, the fact that there's something called recombination that shuffles your chromosomes in every generation. If you go over several generations, you end up with chromosomes that are, in any given population, rather uniformly shuffled up. So there's an expected background pattern of shuffling. Now, the power of whole genome sequencing is you can look across the entire genome for deviations from this kind of random background pattern of shuffling. It's like looking at the pattern of beads on a string. And what you're trying to find is a region where the beads are too similar to each other. In this case, we're looking for evidence of positive selection. So in this region, you can see that that pattern is anomalous statistically. And so something must have been happening there. Well, if you apply this to actual genomic data, you end up with some really interesting insights. This is the application of that methodology using a statistic called IHS, the Integrated Haplotype Score, around the lactase gene. Lactase produces the enzyme that allows you to digest the sugar lactose in milk. All mammalian babies are born with that ability. Almost all of them lose it around the time they wean. So in the case of humans, around the age of 2 or 3, we lose the ability to digest lactose. And everybody, by default, becomes lactose intolerant, except in a couple of populations who, in the last 10,000 years, started to farm and domesticated cattle, sheep, and goats. And milk, it turns out, is an awesome protein source because you don't have to kill the animal. So you've got a continual source of clean, healthy protein and fat on the hoof. And you can make it into cheese so that it lasts a long time. You can store it. It lasts longer than raw meat does. So it's a fantastic food source. There was strong selection, it turns out, on populations that domesticated these animals to retain the ability to digest lactose into adulthood. And so if you have Western, Northern European ancestry today, you're carrying a mutation that originated probably somewhere in Bulgaria around 7,000 or 8,000 years ago and has been spread throughout Europe because it provides the adaptive advantage of allowing you to digest milk. Somewhat lower frequencies in the Middle East, lower frequencies in India, but it's all the same mutation. And it's spread throughout that entire region in the last 7,000 or 8,000 years because of this adaptation to a new food source. We see the same thing, same gene mutated, different mutation, completely independently, in sub-Saharan African populations that herd cattle, groups like the Maasai. They're also lactose tolerant into adulthood, but a different mutation, driven to high frequency by selection. We kind of knew about the lactase story, but the cool thing about having access to genome-wide data is we can scan the entire genome, and we see all these peaks where selection has occurred. Each one of these is a really cool story, that how and why. SLC45A2, that's the gene that confers lighter skin and fair hair. Very, very strong selection on that in European populations. A related gene out here, HERK2, blue eyes. You have a mutation in that gene if you have blue eyes, probably driven to high frequency due to something Darwin called sexual selection. It's an unusual phenotype. People found it attractive. It spread far and wide, much more widespread than you would expect by chance. Genes involved in the immune response, probably driven by the domestication of animals. Animals coming into family scenarios, kids are exposed to animals, new diseases. Most of the infectious diseases that we vaccinate for these days came into the human population in the last 10,000 years because we domesticated cattle, sheep, and goats. So all these really cool little stories that have come out of having access to massive quantities of whole genome data, which we never anticipated back in 2005. The technology has really driven this forward. The ancient DNA revolution. I'm sure that some of you have heard that you might be carrying around a little bit of Neanderthal DNA. This is due to advances in our ability to sequence literally the whole genomes of long dead individuals, Neanderthals that went extinct nearly 40,000 years ago, and discover new species, the Denisovans, up here. That in Russian is actually Denisova, Denisova. It's a cave in Siberia, only described at the genomic level. The only remains that have ever been found of the Denisovans are a pinky bone and a tooth. Completely new species of hominid. Turns out they interbred with us. As modern humans were leaving Africa between 50 and 60,000 years ago, we encountered the Neanderthals and this other weird Asian species of Neanderthal, that's what we call it as a shorthand, we really don't know that much about it. We encountered them, drove them to extinction. We don't see them walking around anymore, except we mixed with them to a small degree. We got it on with our hominid cousins, such that non-Africans today are carrying on average around 2% Neanderthal DNA in their genomes. So in a way, the Neanderthals do live on in us, in our genomes. And how did we discover this? Well, again, it was with these whole genome sequences. Neanderthals, Denisovans, this is something called a principal components analysis. It's a way of analyzing multivariate data, trying to simplify it, look at eigenvectors. If you think about describing a football statistically, and you start drawing axes to define it as a shape, the first one you're going to draw is lengthwise, so it's kind of oblong, and then you'll draw one that's perpendicular to that, that's a little shorter, and so on. It's a way of describing a cloud of data points. And typically we'll look at the first two principal components in a chart like this. In this case, we've got the genomes of a Denisovan, a Neanderthal, a chimpanzee. When you then add modern human genomes into it, what you see is that the Africans stay kind of central, and the non-Africans are shifted toward the Neanderthal. So again, it's a very subtle effect. It's only about 2% of the genome, but non-Africans are carrying enough Neanderthal DNA that in this statistical analysis, they are slightly closer to the Neanderthals. And you can actually go through, and we have done now, you can go through the genome and pick out the particular spots that came in from the Neanderthals, and they do some really cool things. They affect immune response, they affect our hair, they affect, skin color, and so on. All sorts of really cool traits. This is the takeaway, of course, that the popular press has. Ozzy turns out to actually have one of the highest Neanderthal percentages ever measured. But I mean, this is the power of the ancient DNA stuff, is that, it really catches people's imaginations because it reveals totally unexpected things. And, I've said to many people over the last couple of years, if I were redesigning the genographic project today, I'd probably put more than half of our resources on the research side into ancient DNA work because the insights it's yielding are just so fascinating. And it's not limited to interbreeding with our hominid cousins. Things that have happened over an archaeological timescale, completely revolutionizing our understanding of how ideas, cultures, languages spread around. This is a great shot. This is a transect, a tunnel that was, or a channel rather, that was dug by Dame Kathleen Kenyon back in the early 1950s when she was excavating Jericho. Jericho, probably the oldest city on Earth, dating back at least 10,000 years. So this guy at the bottom is standing at 10,000 years ago. All of these layers, the Neolithic, the introduction of pottery, the introduction of bronze, then the Iron Age, all the way up to the present, the stratigraphy that defines an archaeological site. We can now go through and do that sort of archaeology at the DNA level. We can sample the skeletons that are carbon dated and associated with a particular culture, and we can look at shifts at the genetic level that occur as you see these cultural shifts. So no more hand-waving by archaeologists about, did farming spread into Europe on its own as an idea, or was it literally Middle Eastern farmers who moved in and spread it? We now know that so many of these important cultural transitions were driven by marauding migrants. The Bronze Age, probably the scariest time in human history. Effectively, the entire pre-Bronze Age population of Europe was obliterated by incoming Bronze Age warriors from the steppes of Central Asia, bringing with them Indo-European languages. This is a tree of Y chromosome lineages, and in pink here, you have the Bronze Age. You can see that essentially all of the lineages in present-day Europe trace back to the Bronze Age, and anything older was lost. Massive battles, and this was a study that came out earlier this year. Thousands of people in a Bronze Age battle in northern Germany, skeletons strewn across an entire field. It was a really nasty, bloody time. And DNA is giving us an insight into that. Really, really cool time to be a geneticist. I want to end up by talking a little bit about the work that I'm doing now. As I said, I left National Geographic, and the project really wrapped up last summer, moved down to Austin, Texas, have founded a couple of genomic startups since then. One is dog DNA testing, so it's effectively 23andMe for dogs. It's called Embark. So I was chief product officer of that. But the one that I'm most excited about is the one that I'm now founder and CEO of, called Incytome. I want to take you back a little bit through the history of consumer genomics. If we rewind back to the time when we launched Genographic, which was 2004, 2005, there were a couple of basically academic labs that were offering people the opportunity to test very specific things. Am I descended from Nile of the Nine Hostages, or a Jewish priest, the Kohanim, things like that. Literally a few hundred people, essentially making contact on their own and asking specific questions. But the industry, as such, really started in 2005. And so this is number of people tested. And as I said, we hit 100,000 people with Genographic toward the end of 2005. And it continued to go up in a linear fashion. 23andMe launched in 2007, but their service cost $1,000. So you can see it didn't really have any effect on the number of people being tested. They dropped their prices in 2009, and you can see a little bit of an uptick. But the real change, the real inflection point, came at the end of 2012, beginning of 2013. It took us effectively a decade to go from here to there. The first million people tested. It took a little over a year to go to the second millionth person being tested. And now, genetic testing companies are testing routinely 1 million or more people a year. Ancestry DNA alone, one of the big four that I'll show you in a couple of slides, Ancestry DNA alone tested over 1 million people last year. This is an exploding industry. Now, why is it happening now? Well, I can tell you that back when we launched Genographic, people were kind of freaked out by DNA. They were freaked out by privacy issues. Who's going to misuse my genetic information potentially? They were freaked out by what you might do with the DNA. Are you going to try and clone me and create an army of Spencers that are going to go out and conquer the world like Bronze Age steppe warriors, or they just had no idea. Jurassic Park was real science to most members of the general public. Now I think people have a better concept of like what DNA is and some of the limitations. It's become part of the national consciousness. A lot of companies talk about such and such being in their DNA as part of their advertising campaigns now. It is a part of the national consciousness. People are more comfortable with the notion of DNA and DNA testing. People are also much more comfortable with sharing their most private information. When we were developing Genographic and when we launched in 2005, there was no Facebook or Twitter. Now people are happy to share really scary stuff on social media. Suddenly no one cares about privacy anymore. So I think that's been part of the shift as well. And finally, I think we hit this kind of threshold for word-of-mouth viral spread. It is now fairly likely that if you go to, say, a cocktail party, 30, 40 people in the room, one of those people at least will have tested their DNA now. And they'll often be telling you about some of the cool insights they gained from it. And that's a very important tipping point. Advertising, and I can get into this as a separate discussion, advertising, especially to millennials these days, means less and less. Everybody feels like someone's trying to shill to you. But word of mouth recommendations, a recommendation from somebody you really trust, P2P marketing, really, really important. And I think that's played a big role in the viral spread and this huge up spike in the growth curve for genetic testing. It's kind of like the film industry. The film industry is a little bit of a weird industry because you pump all of this money into the first weekend of release, all this money in marketing, advertising, et cetera, and you expect a revenue curve like this. So you're going to make all your money in the first couple of weeks. And if you don't, the movie is canceled and it goes directly to Netflix. But on occasion, there will be a sleeper film that starts off in a few theaters, and people talk about it, and they really like it. It gets good critical reviews, and so it ends up in more and more theaters. And eventually, it hits that threshold point and really takes off. My Big Fat Greek Wedding, one of my favorite sleeper movies, was exactly like that. And if you look at the total amount of revenue, it actually did better than Twilight did. So there's something to be said for allowing that organic process to take place. But I feel like we are in that kind of, that exponential part of the curve right now, and no signs of asymptoting in the consumer genomics industry. Who's left standing? Well, there are really four main players that account for 99% of the genetic testing today. 23andMe has pumped a lot of money into advertising so people know it, probably the best. We were the first. Ancestry is the biggest. They have this amazing business model where people are able to use the genetic results and tie them into the family tree. They actually take a loss. They might break even, depending on what the price point is, on the DNA test, but they make it up in people buying the subscriptions to the online genealogical information. So it's a loss leader. 23andMe, it's a loss leader as well. 23andMe is not in the genetic testing business. 23andMe is in the genetic data aggregation business. They sell the data onto pharma companies. That's their business model. So family tree DNA, niche market, people who are hardcore genealogists. So these are kind of the big four. But what I'd like to just end with, and we can talk about this more in the precept this afternoon, is what I'm doing next. New startup called Incytome, based in Austin, venture funding out of the Bay Area, launching in spring 2016, developing some really cool products. The thing about all of these experiences right now is you sit down when you get your results back, log in on your laptop or on a desktop, and, oh, okay, there's some interesting information, and then you hardly ever go back to it. It's a one and done model, which is not great from a consumer perspective as the person who's creating the product. You want people to engage with the product, and you want people to feel that they're getting insights that are actually useful. This is, somewhat tangential to everyday life, the information you get from all of these companies. What if DNA could be as engaging as Pokemon Go? That's the vision for the future. And so I'll leave it at that. Don't want to say too much more about the details, but maybe we can unpack some of them in the precept this afternoon. Thank you very much.
Spencer, thank you very much. We're going to open it up to questions. Please remember to wait for the microphone. Quick question from me. When you got your own results, what did you do with it? How did it affect you?
I was not terribly surprised because I knew that I was pretty much Northern European, and that was confirmed by the genetic data. I was hoping for something a little more interesting, something unexpected, maybe sub-Saharan African or Native American. Yeah, I mean, the really cool results are the ones where people do get things back that they weren't expecting. And it forces them to start asking questions among family members, and they'll figure out, oh, there was a Cherokee princess on my mother's side a couple of generations back, that kind of thing. That's really cool. I really enjoy working through those results with people.
Questions?
I was fascinated by the Hungarian discovery you mentioned there. And I was also puzzled because I had always envisioned that Attila the Hun came with mostly a bunch of dudes.
Mm-hmm.
And I would have thought that you would have seen the signal mainly in the Y chromosome in the male lineage, therefore, not in the mitochondrial, but it was only 3% to 2%.
Yeah, there was an excess on the Y, probably not statistically significant at those numbers. But yeah, you're absolutely right. It seems like there was an en masse migration, much like the Polynesian migrations, where you literally, if you think about what the Polynesians did, they weren't just setting sail to the next island. They brought their entire way of life with them. So they brought breeding pairs of pigs and dogs, air-layered breadfruit, very hard to propagate this plant, all this really complex agricultural baggage. And of course, enough people to create a viable society on the next island. I think some of these migrations have been like that. And to a certain extent, yes, the Magyar migration seems to have been that way too. Yeah.
Can you also talk a little bit about genetic testing for medical purposes? Is it, spiking up like this part of it?
Genetic testing for medical purposes is a growth industry, and a lot of people are doing it. The utility, depending on what you're testing for, is marginal, unfortunately. People ask me all the time, should I do a 23andMe test, or there are other tests that are much more medically focused that you have to order through a doctor? And my response is usually, unless you have a family history of something, if you are a healthy person in your 30s, 40s, 50s, it's probably not going to tell you anything you don't already know. In part because we haven't made the discoveries yet for the common diseases. Type 2 diabetes, really, really complex. We know there are a lot of environmental factors, also a lot of genetic factors. Probably hundreds of genetic loci that have tiny individual effects, and therefore very, very difficult to detect. Sequencing is going to help. When we get to millions of DNA sequences, we'll be able to tease that stuff out of the data. But any individual will be carrying probably a similar genetic load for those diseases. Where the medical testing is most important is if you have a family history. So if there's a family history of breast cancer, hell yes, test BRCA1 and 2 as soon as you can. If you're young, the vision for the future is every child's going to be sequenced at birth. And as we discover new things, you'll be able to pull down new information. I mean, this new company, we're working with a company out in California called Helix, which is a spin-off of Illumina. What we're trying to do is basically build the app store for consumer genomics. Because we know that there are going to be new discoveries in the future, but your basic sequence will never change. That lives in the cloud. You pull down the data as it becomes available. So that's the insight. It's much more valuable early on in life than it is for someone who, in a way, has run the experiment. If you're in your 40s, you already know pretty much what your health is like and, what to do to keep yourself healthy. The younger you can test people, the more utility it's going to have. Except for these things that are highly hereditary and that therefore tend to run in families. If you're adopted, yes, I think it's useful because you don't know your family medical history in that case.
That was a wonderful talk.
Thank you.
3 related questions. How far back in time can we recover useful DNA? Is that the same across all species? And what is the prospect for extending that further backward?
That's a great question. It depends on how the DNA was preserved. So if you're talking about something sitting in a puddle on a tropical island in Southeast Asia, a few months. If you're talking about something frozen in the tundra of Siberia, we've been able to get DNA out, that's hundreds of thousands of years old. And so it really depends on the state of preservation. The limit right now for human DNA is somewhere around 50,000 years, 50 to 60,000 years. I think probably Svante Pääbo and Eska Willerslev, who are the leaders in ancient DNA right now, one of whom is at the Max Planck Institute in Leipzig, the other is at University of Copenhagen, about to move to Cambridge University. I think they probably more than anybody else would have an insight into that, and I know they're pushing to go further back. There are a lot of interesting finds in paleoanthropology that have come out in the last couple of years, and it would be great to get DNA sequence information from them. It'd be great to sequence Erectus DNA. I mean, I know Svante's working on that.
I think it's a Michael Crichton, but I've sort of seen this scenario, and I think I've read about it, where you can take DNA and inject it into a cell today. And is there a way that by being able to go back 50,000 years, whether it's humanoid or other species that are now extinct, where there's a possibility of actually bringing back extinct species?
Yeah, de-extinction. It's actually a field of study, and there are conferences held every year. Stuart Brand, who created the Whole Earth catalog and was involved in a lot of the early days of the internet in the 1980s, he and his wife, Ryan Phelan, actually are the heads of the De-Extinction Society, and they have regular meetings with geneticists. Craig Venter is tangentially involved in this. It's certainly feasible to do. Probably, I mean, there is a project actually going on resurrecting the woolly mammoth, the Siberian mammoth. Instead of actually trying to recreate the entire genome, though, what they're going to do is use the Indian elephant or the Asian elephant as a scaffold, and they're going to make specific changes using CRISPR, which is a new genetic technology that allows you to edit in a very specific way the DNA. They're going to introduce the stuff they've sequenced from the mammoths that makes them mammoth-like. So the woolliness and the size and everything else. So they'll create a facsimile of a woolly mammoth, and it'll be in a park, and you'll be able to go to Pleistocene Park in Siberia and see these things wandering around. It's going to be really awesome. In terms of like literally creating an entire genome, we're not there yet. Craig is working on methods to do that, effectively creating synthetic life. So he's created a minimum viable cell, the smallest genome that allows it to survive. And that's teaching us a lot of the things that we need to know about how genomes work, how all the genes work together. And then you can move on to more complex genomes. Anything as complex as a Neanderthal is going to be, 20, 30 years in the future. But yeah, in principle, it'll be possible.
Well, I was going down the CRISPR path too, with CRISPR getting as inexpensive, the home kits and everything else. And
Yeah, that's a little scary, don't you
yeah,
think?
I was just wondering how scared you are about the genetic engineering, because in the wrong hands in certain nations and such, creating the perfect soldier, creating the perfect whatever. How far along is that, and does that scare you?
It does scare me because, I do believe that there are unintended consequences that happen with any sufficiently powerful technology. I'm not saying we shouldn't do research on it. There have been people who've called for a moratorium. George Church, on the other hand, believes we should be actively engineering new humans, a Harvard geneticist. And I've actually debated him a bit on that. I think it's an incredibly powerful technology that, holds the promise of curing a lot of human diseases, really nasty diseases where we understand the cause, but we can't do anything to treat them. Muscular dystrophy is a great example. If we could just go in and snip out the bad mutation and stick in a normal piece of DNA and cure some child that's not able to walk, that would be awesome. The worry, as you say, comes when you start to apply it in other directions and take it, in your own hands to kind of guide the hand of evolution. So I'm wary of widespread application, but I think research should be allowed because I think it's so important that it merits that.
It was fascinating. My question is, I'd like to hear your thoughts on the DNA sequencing that they're doing up on the International Space Station. If you think there's great progress, or I'd just like to hear your thoughts.
Yeah, I mean, it's a gimmick. Quite frankly, the technology is really awesome. The technology that we use in DNA sequencing, the basic technology, really hasn't changed at all since Sanger won his Nobel Prize for discovering that if you take a double-stranded DNA molecule and break it apart by heating it or adding a base or something like that, and then you use an oligonucleotide that sits down that's complementary to one of those strands in a certain location, and then you extend that chain one by one and look at what the nucleotides are as you extend it, you can determine the sequence of the DNA. And that's still what underpins everything we do in DNA sequencing. Even next-gen stuff is sequencing by synthesis. It's just a miniaturized, very complicated version of that same biochemistry. What Oxford Nanopore is doing, and those are the little mini-on sequencers the size of a USB drive that were taken up into space, is they're doing a completely new approach, which is you take a piece of DNA, a strand of DNA that's in solution, you attach something that makes it attracted to something on the other side of a membrane that has a little pore, little bacterial protein pore, and it effectively gets pulled through as though you're pulling a string of beads through a narrow little opening. And as you pull the individual nucleotide bases through, they resist being pulled through that narrow opening. And the amount of resistance depends on which nucleotide is there. So if you measure it very carefully, you can go and know what the sequence is. That's really cool technology. And that's where we're going to get to probably zero cost for sequencing genomes, because the prep is so simple, the process is simple enough that literally you plug this thing into a laptop. They have a version that's coming out next year that you plug into your iPhone. So DNA sequencing, potentially ubiquitous. The error rate is really, really high right now, and the coverage isn't great enough to do an entire human genome, but for microbial genomes, pathogens, they've actually applied it in Ebola areas in Africa where you can do, strain diagnosis in a doctor's office by sequencing. Incredibly powerful technology.
Dr. Spencer Wells, thank you
Thank
for an
you.
extraordinary hour.